Hey everyone, this is Dr. Marie McNeely welcoming you to episode 848 of the People Behind the Science podcast. Today I'm joined by our guest, Dr. Bryan Cassone. And listeners, I want to take a moment to thank you for listening today and to give a special shout out to everyone who has subscribed to our show or left a rating or review. We love getting to hear more about what you think about the podcast. And we have a great episode on the way for you today. So get ready to meet another one of our amazing people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest scientist, Dr. Bryan Cassone. So, Bryan, welcome to the show today. How are you?
I'm doing very well. Thank you for having me. It's great to be here. I appreciate it.
Well, thank you so much for joining us, and I'm excited to learn more about you, your career, and your research. So let me take a moment to tell our listeners a little bit more about your current position and how you got there. So listeners, Bryan is a professor and chair in the Department of Biology at Brandon University in Canada. Bryan received his bachelor's degree in biology, specializing in environmental science, from the University of Western Ontario. He received his master's in integrative biology from the University of Guelph and his Ph.D. in biological sciences from the University of Notre Dame. After completing his Ph.D., Bryan conducted postdoctoral research at the Corn, Soybean, and Soft Wheat Unit in the United States Department of Agriculture. And next, he was a CAPS Herta camera Gross postdoctoral fellow in the Department of Plant Pathology at Ohio State University. Bryan joined the faculty at Brandon University in 2015. And Bryan, of course, we want to get to know you as a scientist during our conversation, but also more generally as a person. So could you give us a glimpse of life outside the lab and what you like to do with your free time?
I watch a lot of football is what I do. I like to travel a lot. I generally go to quite a few places every year.
Well, tell me about this interest in football. Do you have a particular team that you watch or do you play yourself?
I used to play football. I used to wrestle back in college, but right now I just watch a lot of college football, watch a lot of NFL football. I guess the sports I do now, it's more, I do a little bit of swimming, do a little bit of weight training, things like that.
Well, Bryan, it's wonderful to hear more about your life outside of science and some of these things that you like to do to occupy your time when you're not working. But I'd love to talk about your scientific side as well. So how do you describe your work to people who are outside of science or maybe just outside of your particular field?
I'm kind of a generalist molecular entomologist is what I would call myself, because we do a variety of different things. We work on agricultural pests. We work on medical pests, as well as just fun research, such as the waxworm, the plastic eating caterpillar. So I don't have one passion. So a lot of scientists, they either focus on one area and become a real specialized with that. And then there's other ones like myself who are a little more generalized. And there's just too many cool things in science for me to stay focused on one area.
Oh, well, I second that. And I think your approach is great. I think once you develop expertise in specific techniques or approaches, they can be applied to a wide variety of different questions. And I think you can absolutely build a successful scientific career that way. And I think working on a wide variety of cool science projects can be particularly motivating. But I'd love to talk a little bit more about what motivates you and inspires you. And I think having these little quotes and sayings can keep you going when times get tough. So, Bryan, do you have a favorite motivational quote or something that you keep in your mind to get you through a tough time?
I don't really have a quote I use. I just put everything in perspective, right? Like my work is my job. You're always going to have highlights and failures, I guess. So a lot of times I might not get a grant. I want to put a lot of time into it. A paper might be rejected 3 times from journals before it gets in. So I just don't let these things bother me. You just keep trucking on and see the good things that are happening as well. That's kind of what I focus on. I dwell on the things that are going really well, right? Because you're going to get peaks and valleys in science. It's just how it is with every scientist. So I focus on the peaks instead of the valleys. And generally, a valley will become a peak eventually.
Well, I love this sense of optimism. Is this something that comes naturally or did you have to work to develop this?
You get rejected a lot in science, so you eventually get really good at it. you're constantly getting evaluated. You're kind of, at least as a professor, you're getting student evaluation, then you're getting evaluated for grants, you're getting evaluated for papers and all that. So you just get used to it and you see the good in it and you see the positive. And when it becomes positive, you really want to savor those aspects.
Well, I love that. I think this idea of searching for that silver lining, even when times are chaotic or where there's a lot going on, there's a lot of this judgment that happens in science can be particularly valuable and can help you stay on track. And I think as a scientist over the course of your career, you might encounter people who help you stay on track or help you kind of reach that next level of your career. So when you look back on your own experiences, Bryan, are there particular people who really served as role models or mentors or who really inspired you?
I've been lucky, particularly with my master's degree. I had a really supportive advisor. I came in with minimal science background, so she was quite supportive. Dr. Bolning at Guelph was really key for me staying in science. And then during my postdoc, I had a really good advisor as well, Dr. Redenboss. She was wonderful and really helped me with improving on paper writing and the scientific communication aspect. And since then, it's been students. Students have been really quite inspirational to me in terms of their progress and their ability to come in with minimal experience and then really become good quality scientists with their own ideas. It's really nice to watch. And it's been really helpful to me. It keeps me passionate about the science.
Absolutely. I think you can draw inspiration from all of these different professional relationships that you develop as a scientist. And it sounds like you had really excellent mentors at these different training stages of your career. And I think these can be critical for helping you pave the way for your own career path. But let's maybe take it all the way back to the beginning. Bryan, can you share with us what first sparked your interest in science?
I didn't have one of those stories where I was just enamored by bugs when I was 4 years old.
Mm-hmm.
I went to university and I had originally thought I'd become a pharmacist. And then halfway through, it just didn't seem like my type of fit. So I did a honors thesis with a professor at Western University. I really enjoyed the independence aspect of that and doing something that's never been done before. So from there is really when I shifted focus and thought that research would maybe something that is in my future. And I got lucky. I got a pretty good position as a master's student and went from there.
Absolutely. And I'm curious, what initially got you interested in pharmacy? Why did you think, okay, this is what makes sense for me to be?
It just seemed like a comfortable job. That's really the extent of it. It just seemed like a good job, a high demand job, a job you can do anywhere. But that's kind of at the same time, the thing that made me go the science route. I guess I like things a little more erratic.
Definitely. And I think biology is the place to be when you're looking for organized chaos, right?
Yeah, that's a good way of putting it.
So let's talk about those early experiences. What then about biology captivated you? How did you decide to make the switch and that, okay, biology is the new life plan?
I've always been the type of person who watches a lot of nature documentaries. So I've always enjoyed animals. I've always enjoyed life sciences. And you take a variety of different classes in your undergraduate career, and there's just so many fascinating things. I think from early on, it was just life sciences was the one area of science that I wanted to focus on for my education and certainly, I guess, career afterwards.
Well, that makes sense. And as an undergraduate, did you get involved in research at all, or did that come later in your master's?
No, just at the end, I did an honors thesis research on birds in my fourth year. That's when I really started. I found a professor who was taking students, a new professor, and I applied for it and it's not a master's degree. It's somewhat parallel to a small master's degree where it's a 1 year of research in a particular lab where you do a small thesis associated with it.
Definitely. And then how did you make that transition from studying birds to studying what you're working on now?
The one thing I wanted to do with a master's degree, I wasn't as focused on the organism at first. It was more the techniques, like getting that type of experience is what I was looking for originally. My master's actually on crabs.
Oh, cool.
Which were really cool. I had a great time working on them. So the genetics of crabs is really what I did. And that took me down the path of invertebrates.
And then as you were approaching the end of your master's degree, how did you make that decision to go forward and get the Ph.D. versus go out in the job market?
I never even thought about going to the job market. I really enjoyed a master's. I enjoyed the flexibility in your schedule. I enjoyed just doing something no one's done before. I've always been into multitasking, and it makes you do different projects at the same time. Your day's always a little different. So after I was in a master's, I was fairly convinced I was going to do a PhD if I was accepted into the program. So I applied to several different places, and Notre Dame was a really good fit for what I wanted to do. for the type of research they were doing there. And it wasn't too far away from my home, my family and friends. I'm from close to Toronto, so it wasn't terribly far.
Absolutely. And I think the big decision point comes at the end of your PhD where you're sort of trying to figure out what to do next. And you actually came to the United States for a postdoc. Can you talk a little bit about what prompted that decision and how you ended up working for the USDA?
I did my PhD in the States. I had a good experience with that. And it's one of those things where it is a competitive job market for postdoctoral fellows and researchers. There's only a certain number of jobs that are ones that would be your expertise or your science would fit. So there were a few USDA jobs I was looking at, as well as a couple of research labs. They were primarily based in the US just because of research fit. And I chose the USDA over a couple other options simply because it was a really good project. I knew I would enjoy doing, I found really interesting, good location, at least for me at the time, and turned out to be a good decision in my opinion.
Absolutely. And I think for a lot of trainees out there, they might be familiar with kind of what an academic lab environment looks like or what it's like to work in one. What was it like working at the USDA, this more government laboratory setting?
It wasn't too different relative to other USDA groups, simply because it was part of the Ohio State University, or the USDA lab was in the same building. So I had a lot of exposure to Ohio State labs. So academia was kind of mixed with government. It was a kind of a satellite campus. So it was sort of integrated really well. So it didn't feel as much like government as it did as just another academia department.
And then I think another huge decision point comes after the postdocs are completed and you're ready to jump into the faculty job search. Can you talk about what that experience was like for you and how you ended up at Brandon University?
I think I went on 4 or 5 job interviews for a tenure track faculty position, and all in the US except for this one in Canada. When you go for these job interviews, it's something very different than what you're used to, because you do, in most cases, over 2 or 3 days, you do 3 different talks. You do a research talk, a teaching talk, and a chalk talk, typically, as well as multiple interviews with faculty and so forth. So it's not something you really, at least for myself, I wasn't overly prepared for at first. And so it took a little bit to know what I was doing. But ultimately, I received a couple job offers around the same time. And I chose Brandon University because I'm Canadian. I like the funding structure, and it was nice to come back to Canada. And it's a liberal arts school, which I like. It's a little bit more master and undergrad oriented, which I thought would be a good fit for me early on. But yeah, it's a really interesting way to look at things, because at one point you're looking for academia jobs and you're thinking, maybe I should go into industry. So it's a time when you're really making a big decision in life. And I don't know, Brandon University just seemed to have what I wanted in terms of flexibility as well. They were happy to let me not just work on one specific research area. They gave me a lot of flexibility, and it seemed to just fit me really well, despite not knowing where branding was before I got here.
Absolutely. I think that freedom that you mentioned to be able to pursue the projects that you're most excited about is a deciding factor for a lot of people when they're weighing academia versus industry, where things are kind of more set on a particular agenda.
Absolutely.
So it sounds like you found a great fit there at Brandon University. I know your lab is doing a lot of different cool projects. So let's talk about your research some more. Can you give us a description of a project that you're working on right now that you are just the most excited about, Bryan?
The waxworm is one that we've been pretty interested in, which essentially is a caterpillar that degrades plastic. So it consumes plastic and is able to break it down within 24 hours of ingestion, which is remarkably fast. So we've made some pretty good progress recently on figuring out how they do that, as well as how we can harness it for more large-scale plastic remediation. So that's one area that I've been pretty excited with. Another one is mosquito research. We're doing a variety of projects to understand how repellents work in mosquitoes. So DEET's been around for decades, and the actual mechanism of action, we still haven't figured out. So we've got some pretty, I think, interesting work coming out soon with that, as well as certain hormones and how they impact our fecundity with the larger goal of mosquito control. So those are the two I'm pretty excited about right now that we've been working on. And we have some relatively new research coming out in the coming months.
Well, I love this waxworm project. I think this is fascinating, the idea of being able to break down plastics, because this is obviously a huge global problem. Can you talk a little bit more about how this type of solution could potentially scale to really have an impact on the global plastic problem?
I feel there's 2 potential ways that this could happen, where we could actually scale it to a point where you can make a significant contribution to plastic remediation. One would be through just large scale rearing of these caterpillars as part of a circular economy. So what do you do with all that additional biomass when you're raising so many of them? Can you use them in aquaculture? Can you use them in other things to extract economic benefit? So that's one way. And I've been a little bit more optimistic on that approach than I was previously, simply because of the efficiency of it has been improving. And we've been, at least us and other labs, have been getting some pretty interesting results that suggest that this could be feasible. And then the other possibility, which is probably the most realistic, is re-engineering the plastic biodegradation pathways outside of the animal. So if you can figure out the exact or precise enzymes being produced by the animal, the precise enzymes being produced by the host microbiome, so we think the gut bacteria, gut microbes are involved, you can figure out the precise ones for that, take them out of the animal for re-engineering for large-scale remediation. It is another approach we've been looking at, and as well as other labs.
Very interesting. And I think when a lot of people think about plastic breaking down, it sort of has a negative connotation almost, because you start thinking about microplastics immediately. Do you know, I guess, how these waxworms are breaking down the plastics and whether these downstream products are safe?
We know that there's nothing toxic in the downstream products. There was originally the thought that there was ethylene glycol, but there has been no evidence to suggest this. Now, the micro or nanoplastic aspect is something we've been working on. Preliminarily, we haven't found anything, but we can't say with full confidence yet that there's complete biodegradation. For the types of approaches that have been used so far, they haven't uncovered any, but there are other ways to do this that we've been looking into.
Definitely. Well, these sound like very exciting projects. Let's talk a little bit about this mosquito repellent work as well. You mentioned looking into different hormones and looking into just the mechanisms that these different mosquito repellents work to keep mosquitoes away from us. So what are some of the biggest unanswered questions when you're looking at this project?
We've been looking mostly at citronella oil and DEET. So both of those are fairly common repellents. DEET is the most used repellent in the world. It's been this way for a long period of time. But we know that they repel mosquitoes. We just don't know how. So if you can figure out the precise, what we call molecular targets, so what's happening at the genome level in a mosquito, you can presumably build new or create new repellents, which is kind of the ultimate goal with that. So there's been a lot of speculations, a lot of preliminary results for this, but the actual causal mode of action hasn't been fully established yet. So we've been doing some work on that to try and figure out what's happening. I can't say too much right now, but it looks like there's a multimodal effect. Some people have insinuated this, but we think we figured out some interesting targets that are actually working with DEET and citronella and how they actually induce the repellents.
Very cool. Well, listeners, we'll have to look for published papers coming out on this topic soon from Bryan's lab. And I think there's still so many unanswered questions in science, but answering them often isn't easy. I think there are struggles, there are failures. There's a lot of criticism, as you alluded to earlier, that goes into your life as a scientist. So let's talk about the difficult times. Bryan, I know these stories often don't get to see the light of day. You often only hear the successes, but do you have an example of a major failure, a time where you really struggled with something that you could tell us more about?
I've been pretty fortunate that often if a paper doesn't get into a particular journal, I can usually get it in somewhere that is a better fit for it. But there's been certain papers like the first waxworm one, which had a significant media interest. We went to several other journals prior to it being accepted, and they told us it was too niche. Nobody cared really about the waxworm in it degrading plastic. We were eventually able to get it published, and then it gained quite a bit of media traction from there, which was very different than what our editorial reviews were at other journals to begin with. A lot of times with grants too, you'll not get a grant. You're not even sure really why. Sometimes will get really positive reviews from reviewers, and then it just doesn't get funded. And you don't really get the specifics on why this is the case.
Right.
But it's a competitive process. So you can only fund so much research. And there's been a lot of projects that at least I thought were really good ideas that go nowhere because you just can't get the funding for it. Even the waxworm, we don't get a lot of funding for that relative to some of my other projects. So it's just what a funder is looking for at a certain time, I guess.
Absolutely. I think recovering from bad news is an important part of being successful in science. So how do you recover? How do you bounce back, Bryan, after you get one of these rejections, whether it's a grant that wasn't funded or a paper that you were really excited about that was rejected?
You're always a little disappointed at first, but I think when I first started, I was a lot more disappointed. Now it's just, I'm at the stage in my career. So even early on, you have a lot more pressure because you're looking for tenure, you're looking for promotion. And just to begin with, it takes months in most cases for a paper to go through the review process. So you need to get their currency for your life and what you're doing in science. If you fail out and don't get tenure, you can't do any research anymore. You have to get a different job or go into industry or something like that. So there's a lot of pressure with that. So if you don't get a grant that you really think was a good idea and could be pretty meaningful contribution to whatever field it is, it can get you down a little bit. For me now, I'm fully tenured. I don't worry as much about it. It's just sometimes it's like, oh, that's too bad. I would have really liked to do this project. Maybe I'll find another outlet for this in the coming months. It's okay. It's not a big deal. You don't get a paper in somewhere. I mean, I will say I've seen some bad reviews or just some reviews that I think are inappropriate from reviewers for research. I haven't had a lot of those. So a lot of times they'll give you some sort of constructive criticism. So whenever I review a paper, I don't criticize. I try and give constructive feedback, which I find more and more reviewers are doing. So that's good. And then you can take that and maybe hone the paper, resubmit somewhere else, or just resubmit to another journal that maybe has a better scope. So for me, it doesn't really do much to me anymore. You just keep going. If you think it's a great idea or you think it's a great paper or a really good paper, you can't get down on it. You got to trust yourself and focus on trying to get it out there or get the money for it so you can do that project a little bit later.
Absolutely. It sounds like you've become immune to the pain of rejection.
Yeah, I mean, that's what I tell people. I'm like, oh, you're constantly getting evaluated, even with students too. Mm-hmm. We get student evaluations for every single course we do, right? So it's just, I know some people have some really bad evaluations, right? You just can't let it get you down.
Absolutely. You got to keep a stack of those nice evaluations by your desk.
Yeah, they're usually really quite constructive. It's usually not bad, but there's certainly a lot of horror stories about them.
Of course. Well, thank you for talking about some of the difficult elements of your career. And I think you're absolutely right. The early career stages when you're trying to establish yourself can be difficult. And when you make it to this point where you're able to be a little bit more free and not worry so much about each individual rejection can be refreshing. And you've had a number of successes along the path to get to this point. So I'd love to talk about some of your successes, Bryan. Do you have a favorite success story, whether it was a big one or just a small but particularly meaningful one that you'd like to share with us?
I think my biggest win was the very first grant I've submitted. I came to Brandon University and I had some soybean background, but I had never actually wrote a grant in my life before, like a full grant on my own. I've contributed grants when I was a postdoc, but never led a grant. And the very first grant I submitted, it was, can't remember how much it was for, maybe 150,000 or something like that. We submitted it to a soybean commodity group, which was the predominant soybean research. So they do, obviously it's production, but they allotted some money to research every year. And I had no thoughts that I would ever get this grant. So it was surprising. It was really a great feeling when I had received the funding for that grant because I had not expected it. They were very favorable in their reviews of the study and the interest in what it could do for soybean production. So that was really one of the more memorable moments, I guess.
A great introduction to grant writing, which I'm sure was probably a little bit downhill from there after that initial victory, right?
Yeah, you can't bet 1,000 the entire career, but... Yeah.
I love that. Well, can you tell us a little bit more about this grant? What was your proposal?
So this was right when next generation sequencing was coming out, which is just sequencing all of the RNA and DNA in a given organism. So it hadn't been applied to plants for pathogen detection. So we essentially applied that for both detecting pathogens as well as figuring out the genes involved in the invasion process. So it was good. We discovered pathogens that had never been reported in Manitoba and really helped out with the control and certain areas to control certain pathogens that may need control, because prior to that, it was mostly visual, which was highly inaccurate. So we were able to kind of give growers and the commodity groups an idea of what really is in their field and at the prevalence that they are there.
Interesting. And this grant, it was the first try, it sounds like.
Yeah, award on the very first try, which was good. And it went well. It went successful because we didn't know exactly how to do it at the time. So it was kind of a newer approach for what we were doing. We had never applied this to this type of system. And so it worked, which was good. That was my concern, is it wouldn't work at all. And we would have just wasted money, and nobody wants to do that.
Of course. So how did you celebrate when you first got the news that it was funded?
I think I had a couple drinks, had a great time. Just had a good day. Just had a good day, a good week. Probably went out for dinner, that type of thing.
Fantastic. Well, I think these successes in science are important to take a moment to celebrate, as you mentioned earlier, because oftentimes there are a lot of failures along the way, or just a long journey to get to the point where you have a success to celebrate. But we like to encourage our listeners to not just think about their own science, but to also think more broadly and to read more broadly if they can. So we love recommending books to everybody on our show. So do you have a favorite book, Bryan, or one that you've read recently that you've really enjoyed that you'd like to tell us a little bit more about and recommend we add to our own reading lists?
At this point, I just read scientific articles because there's so many of them I want to read, and it's every day I'm reading articles. So I don't really read actual novels or anything like that. I read textbooks, Principles of Virology. That's a great one. But ultimately, it's mostly articles that I'm reading.
Do you have a favorite book that you've read previously before things got very sciencey?
I've enjoyed a lot of books that are more into the drug scene. David Simon's The Corner is one book that I really enjoyed. It's about the drug epidemic in Baltimore.
Well, we will add this to our book list online for our listeners to find there. And one of the things we sort of touched on in talking about your career path are some of the opportunities that you've had to travel, to go to these different places for the different steps of your scientific training. And I think travel is a huge part of life as a scientist. You get to go to conferences, work with colleagues all over the world, sometimes do field work. So do you have a favorite place, Bryan, that your science has taken you?
I went to Costa Rica to do some work there. I really enjoyed that place. What we did is we did a little bit more of viral detection or viral discovery through some of the water, some of the hot springs and things like that over there. So that was really good. And during my Ph.D., went to Mali worked on the malaria mosquito. So we went to visit a lot of different villages and did a lot of collections there. So that was a really interesting and eye-opening experience for me. And then for conferences, I've been lucky enough to go to a lot of different places, Japan, Belgium in the last year, and just a variety of other places where entomologists gather for research.
Very cool. And this experience going to Mali during your PhD, you sort of threw that in there really quickly. This sounds like a life-changing experience. What was that like for you?
It was good. It was really good. When you're working on a mosquito that's responsible for so much morbidity and mortality, and then you actually get to go there, you get to collect the actual mosquito and see what it does to people, as well as how things are different in terms of infrastructure in these isolated villages where people are coming down with malaria frequently. It was a great experience.
Definitely. So was this one of your first international travel experiences?
In terms of research, yes, it was. I had gone to Europe a few times, but not on the continent of Africa or anywhere like that. So yeah, it was definitely my first research experience that was outside of North America. So I did my master's on crab genetics. So for that one, I basically drove from Vancouver all the way down into Mexico, into Ensenada, across the Baja California Peninsula, and then all the way back to Vancouver. So that was, I guess, my first travel for research. And then that was my first one outside of North America.
Oh, very cool. So what was it like being out there in the field? We'll start with the crab experience, collecting these samples and doing science out there in the wild.
It was great. You go in and you have no idea really what to do because it was my first true field work. And I brought a research assistant with me who was just a friend who was down to go travel for 6 weeks. So we had to go to multiple different places to collect crabs. So we had to figure out how to collect the crabs, which are intertidal zone crabs. So they live underneath a rock. So you just go around to all these places, lift a rock, and then they scurry away if they're there. So it takes a long time to get the number of crabs you need to do your population genetic study because you need a certain number for it to be robust statistically. Mm-hmm. And so, yeah, you don't really know what you're doing. I remember we didn't have a permit. For the first couple of places we went, we were waiting to pick up our permit. So we didn't actually have a permit with us the first couple stops we made. We finally received the permit in Sacramento. We literally picked it up in person. And then the next day when we were out collecting crabs, we got pulled over by the Department of Wildlife and Fisheries. So that would have been a problem if it had happened before we had our permit with us. So it was little things that you don't really think about as much as being significant consequences or significant in terms of your methodology. So it was pretty interesting.
Oh, definitely. And then what was the experience like actually collecting mosquitoes in Mali?
That was interesting. There was a senior researcher there who helped coordinate with some of the locals in the villages that we would collect mosquitoes in. And they would help us with nets. They would take nets and collect them. We were targeting mating mosquitoes. So during the act of mating, so what happens is they swarm together. The males and females will swarm. And then the males will grab a female and mate with her as they fall to the ground. So we were interested in seeing what was happening at the mating in terms of genes and all that. So they would go with their nets during the swarming during the evening and collect them that way so that we'd process them and so forth.
Very interesting. Well, these sound like remarkable travel experiences. And I think going out and working in the field gives you an opportunity, like you said, to experience different things, see new places, experience new cultures as well.
Absolutely. That's one of the perks of the job is the travel and the flexibility of schedule. Those are the two things in life that I really value. And that's the one good thing about being in my job is you get both of those.
Definitely. I think another perk of the job is just the people you get to work with. You mentioned previously, you get to get inspired by your students every day and these other individuals that you're working with. And I think there's often a particular stereotype in the media about what scientists are like and what life as a scientist is like. It's all white lab coats and pristine environments. And I think that's not the case in so many different laboratories and scientists that I've spoken with. I think there's so many funny, creative, amazing people doing science who are interested in a lot of different things, not just science. So we love sharing the human side of science and just talking about some of these fun traditions and funny memories and just quirky personalities that you see. So do you have a story of your own, Bryan, that really showcases this human side of science?
A lot of times in my lab, you wouldn't even know that they're in science. A lot of students or grad students, they just don't personify that type of stereotype. We always do lab activities. We go for lab bowling. We go for lab parties. We'll have a keg at a lab party. So it's just more relaxing and regular people hanging out. It's not just hanging out, talking about science. The last thing you do when you hang out with your lab at an informal gathering is talk about science. You just talk about life and you just act like normal people, really.
Absolutely. And you mentioned lab bowling specifically. Are you an accomplished bowler?
Oh, no, I'm not. I wish I was. I've bowled enough that I should be, but I crack a 140 on a good day or a 150. I enjoy doing it. I enjoy that ball, like lots of little activities.
I think having this kind of fun social environment is important so that people can feel like you have a community in the lab and that you're working together to solve these big problems.
Absolutely.
And I know some of the research we talked about today is quite global in scale. You're trying to answer some of these big, very important questions, but oftentimes there are limitations, Bryan, whether it's funding, technology, just the feasibility and time it would take to do projects. So if we took away the barriers that normally hold you back and you could have anything, what is the one research question that you would want to answer most?
I think in terms of just infectious disease transmission, it's tough because infectious disease transmission, plastic are two things I'm pretty passionate about. And they're doing transgenic mosquitoes and all of that right now. But I think if you could identify a microbe or part of the microbial genome of a mosquito that allows them not to, and they do this a little bit with Wolbachia, allows them not to actually transmit pathogens, that would be something that we've been looking for a little bit because every single mosquito has a different microbiome. And at least for ticks, what we see is there are certain microbes that are associated with ticks that do not transmit Lyme disease, for example, whereas they don't seem to have one present or they seem to have one present. So if you could find that for a mosquito and actually prove that it is involved or it can inhibit malaria transmission, that would be really cool.
Well, I think this is a fantastic question or a fantastic dream project that would, like you alluded to, have global impact. So thank you for dreaming big with us on the show. And we talked about some different aspects of your career. And I think when you're at these transition points in your career, it can be really helpful to have advice. So is there a particular piece of advice, Bryan, that you received at some point that really helped you and you can share with all of our listeners today?
The best advice I've been told was from my dad. Just do your best. That's all you can do. As long as you've done your best, even if it's not quite to the level where you're going to win or get the grant, as long as you feel you've done your best, you put in as much effort as you can, you should be happy with yourself.
Well, I love that piece of advice. And I think that's very helpful in science because oftentimes there are these factors outside of your control that dictate the outcome, but you have control of the effort that you put in.
Absolutely. As long as you've done your best, you have nothing to be ashamed of.
Wonderful. Well, great message for our listeners. Is there any other piece of advice or maybe a last note of inspiration that you would like to share with our listeners at the end of our conversation today?
If you really enjoy your job or you're interested in what you do, you're never going to feel like you're actually working and you're going to work harder and enjoy what you're doing. So I understand that work is called work because it's work. But if you enjoy your work and you get a lot out of it and you're interested to go to the office every day, then you're living life because you spend 8 hours or more a day on work. So if you're enjoying it, it doesn't become as much a task anymore. And it becomes something you actually sometimes look forward to doing rather than not. So I would say if you're not happy with what you're doing, it's never too late to try and take a risk and see if you can have a career or a job that's a little bit more satisfying to you.
Definitely. I think trying to align those aspects of your life, find something that you can work on that you're passionate about or that you're excited about going in and doing every day. I think that can be a huge transformation in your life and improving your quality of life.
I couldn't agree more.
Wonderful message for our listeners. If our listeners want to learn more about you, Bryan, and more about the work that you and your lab are doing, what is the best way for them to do so?
I'm always checking emails. So cassoneb@brandonu.ca. Happy to check emails, answer any questions you have of a meeting, whatever you want to do, as well as Google Scholar. So my research is up there. ResearchGate. So both of those I'm accessible on. So if anybody's interested, I'm happy to talk further.
Fantastic. Well, thank you for sharing those resources, Bryan. Listeners, definitely take a moment to check out Google Scholar, check out ResearchGate, learn more about Bryan's work. And we talked about, of course, some upcoming work that will be coming out soon that you don't want to miss. And Bryan, it's been such a pleasure to have you on the show today. So thank you so much for your time.
Thank you very much, Maria. I really appreciate you having me on.
Well, it was wonderful to chat with you, listeners. Always great to have you here with us as well. We hope you'll join us again for another episode of People Behind the Science.